welcome to the second week of this year's Tuesday Lux seminar, where we invite internal speakers, mainly from our EEB department, to give seminars on their work and research. Today we have Dr. Jacob Berv, a postdoctoral scholar and life sciences fellow in the EEB department, working with Professor Stephen Smith, Matthew Friedman, Dan Roboski, and Ben Winger. Jake received his undergraduate degree from the EEB department at Yale in 2010, where he worked with Dr. Richard Frum. After some time off, he went to receive his PhD in EEB at Cornell in 2019, where he worked with Dr. Berv and was funded by the NSF-GRFP grant. Jake's work lies within organismal biology, especially ornithology, and his primary research interests are in the intersection between micro and macroevolution and studying these domains by combining genomic, anatomical, and paleontological data sets. He has received a number of awards and recognitions, including the NSF-GRFP and Doctoral Dissertation Improvement Grants, and has given numerous conference presentations and invited lectures. So today we will hear from him on the molecular early bursts associated with the diversification of birds at the KPG boundary.
Wow, thank you. You did your research. I think I forgot about a few of those things. So hi, I'm Jake. I've been a postdoc in this department for a couple of years now, but because of COVID, I feel like I'm still floating around in this nebulous zone. But anyway, I'm very happy to be here to talk to you all about this stuff I've been working on, mostly in the shadows for a couple of years now. So this is a big sort of collaborative project, and we're just about to submit this as a preprint, so it should go on this week. So if you're interested in the stuff I'm going to tell you about, just follow me on Twitter or email me, and I'm happy to share the preprint when it's available. So, oh no, this isn't going to work. So I am a systemicist first, and I think about a lot of different scales of evolutionary biology. So I work in molecular evolution, phylogeny reconstruction, thinking about how old things are using phylogenetic methods to infer the ages of divergence among lineages, comparative methods, so how do we use statistics to test hypotheses about the process of macroevolution. But I also have a deep interest in neotropical biogeography, specifically subaceans, which are a hyper-diverse group of passerine birds. And I've had the privilege of working on some systems like these mannequins up here and cotingas. We just published a paper on mannequins recently, which took almost 10 years to do, so I'm very proud of this one. My sort of approach to thinking about evolutionary biology is an approach that basically uses models to think about different kinds of biodiversity. So connecting micro and macro scales with models is sort of the thing that I am trying to do. And I really like this way of this sort of cartoon because it kind of articulates, it kind of describes this process, which is an iterative process that allows us to learn about how evolution works. So we often start, at least with molecular systematics, we often start by collecting sequence data. We then use that data to reconstruct an evolutionary history using some approach, some method. We use what we learn from that to learn about evolutionary processes. We then use what we've learned about evolutionary processes to actually develop better models. And we sort of then feed that back into our sequence or our original data and go around and around. And we sort of iteratively refine our understanding of how evolution has occurred and how it works. And I personally like this kind of approach because it allows me to sort of jump around theoretical and empirical domains and allows me to sort of dip my toes into lots of different aspects of biodiversity science. So paleontology, systematics, computer science, molecular biology sort of all speak to different parts of the stuff that I do. And but today we're going to talk about deep time in relation to these ideas. And the kinds of changes that we often think about or that we're often most interested across deep evolutionary time are these ideas of tempo and mode in evolution. And oftentimes the studies that we read about are asking a question about either the process of evolution, the mode, or the speed of evolution, the tempo. And in paleontology, there's some authors who would argue that modes, the process of evolution is really constrained to either cladogenic or anagenetic modes of evolution change. This is a paper by Jablonski that talks about this in terms of morphological change. But Simpson had a little bit of a wider definition, a more general definition, I think, which included something that he called quantum evolution, which is basically the idea that the mode or the process of evolution can change very rapidly across geologic time. And so that there may be certain times, intervals of time through history where you see these big jumps in how evolution is working, how evolution is operating. He talked about this in the context of, so he's got a cartoon from his book, Tempo and Mode in Evolution, where he talks about sort of an explosive phase of evolutionary change, followed by sort of evolutionary stasis. And so whether or not this is the same thing as punctuated equilibrium is sort of not the point, but but anyway, this is a kind of way of thinking about the process of evolution, right? So we can think of the mode as the process itself. What are the rules describing what kinds of changes that can occur, and the tempo as a sort of a component of the mode, but something that's kind of different. Recently read a paper in Matt Friedman's lab meeting that talks about these terms tempo and mode, which today have different, depending on who you ask, have really different meanings. And so I would direct you to this if you want to dive into that debate. It's a nice chapter, nice review. Or email me and I'll send you a PDF. So why is it super important that we study the mode of evolution? And I'm going to start this next little bit of the talk just by introducing us to a particular way of thinking about how we test hypotheses in evolutionary biology and macroevolution. And I'm going to use this example from Slater 2013 that looks at fossil mammals through time, fossil body masses of mammals through time. So in this example, each little dot here basically represents the body mass of a particular fossil mammal. Here's the, log of mass on the horizontal axis and just time on the vertical. And you can see that basically up through the Cenozoic, up through the Mesozoic, the evolution of mammal body mass is kind of constrained to this sort of interval here. And then there's a big gap. Dinosaurs go extinct, big gap in the fossil record, and then all of a sudden, the present, there's the appearance of modern mammals and modern mammal fossils, and there's this big disparity, big variance here. So how do we make sense of this? I'm not a mammologist, so I'm not claiming that this what I'm about to tell you is like the true story of mammal evolution. I'm just using this as an example to make a different point. But one way to study this is to use, basically to draw from the literature of stochastic processes. And one very typically used approach is something called Brownian motion, which is sort of a random walk through time. And maybe one way you might want to study this is by fitting two models of Brownian motion, one in the Mesozoic, one in the Cenozoic, as an example. And the only thing you really need to know about these models in terms of how they work is that there's one sort of key parameter that describes this rate of diffusion called sigma squared. And it's in units of the trait that you're interested in squared over time. And so in this case, maybe the rate of diffusion is slower in the Mesozoic and faster in the Cenozoic. And so you get a slower accumulation of disparity in the Mesozoic and a faster accumulation of disparity in the Cenozoic. And that might explain this distribution of points that you see over here. So if you were to then actually fit these models to those data, and you were to get, hypothetically, these aren't actually from the paper, this is just an example. Let's say you measured this parameter to be 0.05 in the Mesozoic and this parameter to be 0.25 in the Cenozoic. That means that we can infer, indeed, the rate of evolution, rate of character evolution was slower in the Mesozoic and faster in the Cenozoic. So this is a way of using stochastic processes to test patterns of evolutionary change over time. You can measure evolutionary parameters, you can fit models like this, you can be creative about it. But a question for the students in the audience, is this a good model? Is this a good way to think about mammal evolution? Or is there something that we've overlooked? I'm not, anybody, I'm actually asking for audience participation. Anybody have an idea? Is this a good model? Go for it.
Maybe there's an optimum.
Sorry?
Maybe there's a body size optimum in the
Well, I think you're overshooting.
Oh, sorry.
Yeah.
Thought it was
No, it's a good answer, but it's definitely the right answer, but it's more advanced than what I'm going for. Yeah. Sorry?
I don't know what you want.
Yeah. Well, okay. Well, let's think of an alternative model. Does anybody have any ideas of what alternative models we might want to try, or might want to, in terms of just think about You don't need to know any math or any, and just conceptually, what kind of, do you think that an unbounded process of unbounded diffusion makes sense biologically? No. Anybody? You want to try again?
There's a bound. Body size can't be lower than 0.
Sure, that's a good point. That's certainly a good point. It's certainly not, it can't go below 0. That's a great point. So that's close to where I'm going. So let's imagine an alternative model with a force that's like a bounding force, like a rubber band, that's constraining where the phenotype can go. And let's add one more parameter called alpha, and let's just set it to an arbitrary value of 1. And if you run this model and set this rate of evolution parameter, which is the same as the rate up here, to 0.5, you actually get much less, very tight evolution. So much less variance is accumulated over time by adding this additional parameter that reflects a bounding force on where evolution can go. So right away, you should note that this diffusion rate is much, much higher than either of the diffusion rates in these examples up here. And yet, the variance accumulated over time is much lower. And so this is telling you something about why it's critically important to think about the mode or the process of evolution when you do these kinds of studies. Because in this example, as Slater shows in this paper, this model, where you actually have a slow Ornstein-Uhlenbeck process, which is this bounded process, followed by a fast Ornstein-Uhlenbeck process, followed by a slow Brownian motion process, is a better model than a slow Brownian motion process followed by a fast Brownian motion process. So this tells us that if we fail to have an accurate understanding of what the process generating the data is, we can be very badly misled in our inference of the process. And so a failure to consider how the mode might have changed can lead you to be, to think that evolution might work in a way that it's not. So it also, I think, illustrates how tempo and mode are really confounded by one another. If you have the wrong concept of a mode in your analysis, you can infer the wrong rate. And so tempo and mode kind of play off of each other in that way. Okay, so that part of the talk is done. This is mostly just to get everyone up to speed on sort of the way I think about this kind of approach. So during my PhD, I got really interested in this effect of the end Cretaceous mass extinction on subsequent diversification of most vertebrate groups. So while we know that this event marked the end of the non-avian megafauna, much less is known about what actually happened to those lineages which survived. And especially what happened in the immediate aftermath of that event. So one thing that probably happened to lots of different groups, although the evidence is mixed in some, is this idea of size reduction associated with the mass extinction. And so there is direct evidence in the fossil record of this occurring in a number of different groups. The case of mammals is probably not as clear as I used to think. Thanks, Luke, for pointing that out to me, wherever he might be. But there is good evidence in other groups where this is probably the case. And when I was doing my PhD, I got really interested in how that phenomenon might impact rates of molecular evolution in birds. And we published this paper where we were asking whether or not a changing body size in birds could affect rates of molecular evolution in such a way that it would bias our ability to actually test macroevolutionary hypotheses. And we used molecular clocks to do this. So this is not the point of the talk I'm giving today, but this work made me think about how important models are in the way we study evolution. And basically the bottom line with this was that it really depends on whether or not you believe the fossil record or not. If you believe what the fossil record tells you about the evolution of birds, then that also induces some specific requirements about what you have to believe about molecular evolution. And so given that the fossil record is imperfect, you have to sort of make some decisions about what you want to believe. But when I started this postdoc, I got really much more interested in this mode question and whether or not there's any evidence of mode changes in how the genomes of birds were evolving associated with the end Cretaceous mass extinction. So is there any reason to think this might be a thing? So there are two kind of interesting examples that I point out first. One is this example from Van Est et al. 2014 that identifies a sort of a wave of whole genome duplications in green plants associated with the end Cretaceous. So this has been corroborated in a number of different studies. It's a fascinating problem, and I'm not an expert on this domain, but it seems like this is a good example of a change in the evolutionary process that's driving the evolution of genomes in these groups. So another example comes from birds, actually. It has to do with gene tree species tree discordance. It turns out that over the last 10 years, one of the main things that we've learned from bird systematics is that there's a huge spike in basically phylogenomic discordance, so conflict among individual gene trees as you approach the end Cretaceous boundary. And so something has changed here, right? So like here's right before the boundary, and then immediately you see this gigantic spike in gene tree discordance. So something is, there are population genetic reasons why this might be the case. But as a sort of a precedent for thinking that the genome can be affected in sort of fundamental ways, these are kind of two examples. So in comparative methods, sort of the field of studying quantitative characters on trees, there are a number of different methods that have been developed over the last decade or so that are sort of varying in their level of sophistication. And now we have these approaches that are extremely flexible. So for example, in this method that's pretty cool called PCM fit, allows you to fit multiple models to a tree, where the model can change over the tree, and in a very flexible way, depending on the data and the tree. And the model identifies where on the tree those models have changed. And so a paper published a couple years ago by Josep Ueda called this approach phylogenetic natural history, in the sense of trying to identify where the process has changed, where the process of evolution has changed on a phylogenetic tree. And there are a number of different approaches. Here's just some examples that have very different philosophies on how to do this. And so I wanted to, when I started this job, I wanted to sort of ask this question, but about bird genomes. And so here's the question we're going to answer today, or try to answer. Was the mode of avian genome evolution associated with the end Cretaceous mass extinction? So with sequence data, there are fewer tools available to do this than for quantitative characters. There are a few that exist, there's quite a sort of a, I don't want to say rich, but mature literature on this topic that have been, so going back to Peter Foster's work in early 2000s. So there are a couple of tools we can use to actually ask this question, but most of these don't work with phylogenomic scale data, and they and they're not quite as flexible as we need for what I would like to do. So it turned out that Stephen and some colleagues are working on a new method called Janus, named for the Roman god of transitions, that will do pretty much exactly what we need to do to answer this question. So this paper is now available online as a preprint. So what can it actually do? Given a phylogram, so a tree with branch links and substitution units, it will identify where these key parameters of molecular evolution, so the evolutionary rate matrix or the equilibrium base frequencies, may have shifted on the tree in a discrete way. And it'll identify basically convergent or divergent or convergent patterns of molecular evolution on a tree, and it'll spit out these parameter estimates for each of these regimes that it identifies. So this is really a pretty amazing tool, and it works really well. And so if you're interested in these questions, I encourage you to check it out and play with it, or to contact Stephen or me, and we can talk about it. So this is what it can do. What kind of data are we going to use with it? So I decided to, rather than trying to reinvent the bird tree wheel again for the 10,000th time, I decided not to do that. And we're going to use a published phylogenomic supertree of birds that was published in 2019. There's a couple of reasons why we're doing this, but the actual sequence data that we're going to use is a reprocessed data set from this Promedol target capture data set that I was involved in producing 6 or 7 years ago. So we did this, and we were able to generate about 800 new, it's a new data set. We reprocessed it from the raw data, 800 phased loci with less than 10% missing data. So it's a nice new data set. And the main collaborator on that part of the work is Sonal Singhal, who was a former postdoc here in this department. And I just want to put this out to just basically just to emphasize her contribution. We ultimately were able to generate from this original target capture data set a really nice data set that's been parsed out at the data type level, so we identified exons, introns, and UTRs and separated them into separate data sets, which is typically not done for target capture data sets. So anyway, so we also got mitochondrial genomes from this too. So working with Rebecca Kimball and Ed Braun, we were basically able to generate whole mitochondrial genomes from this sort of second generation-ish target capture data that was originally collected almost 10 years ago. So in general, what this means is that we can really, I think that we're now sort of in a phase where we can like kind of look back at some of the first generation target capture data sets from 5 to 10 years ago, and we can reassemble them and like triple the size of the data set and get out organelle genomes and all sorts of stuff that we weren't able to get before. So yeah, I guess I would just encourage everyone to try to use the resources that we have. Okay, so but with respect to Giannis, what are we going to do? Or what are we going to ask specifically? So we're going to simplify the problem a little bit and just focus on this vector of equilibrium base frequencies and ask where on the tree is there good evidence that these parameters have shifted? So the next couple of slides I'm going to show you I will try to walk through them slowly because they can be a little bit dense, but here's the first major result. So here's what we have for exons. So look at the whole exon data set together, we find 8 distinct regimes across the avian phylogeny. This 8 model optimization receives about 100% of the model weight, so it's very well supported. And in this radar plot, I'm sort of showing the actual parameter estimates from Giannis directly. So each axis here represents sort of the proportion of nucleotide, the base frequency proportions in equilibrium. So it just sort of shows the colors here match the tree. It doesn't really matter that you can't see any of these things. The point is that most of these colors sort of identify really deeply diverging clades in the bird tree. So let's look at introns. We identify 6 distinct regimes in introns, some of which are overlapping with the exons, again with 100% model weight. UTRs, fewer, 4 distinct regimes, but also identifying these really deeply diverging clades. And then mitochondrial genomes, the fewest number of regimes, but again, really deeply diverging clades. So in this case, we only identify basically an ancestral regime, a shift in tinamous, and then a shift on neognates, so the rest of modern birds. And so this is sort of one way of visualizing these data. To sort of summarize those 4 slides, those 4 different data types, I kind of just stack them on top of each other. And this is what you get when you do that. So in this case, I'm just plotting every clade, every single group that is a different color is a group that is identified to have one of these molecular shifts. But sometimes the data types are not exactly the same across these different data types. So the way I've done this is each little square, which you probably can't see, sort of describes the pattern of model shifts in each particular group. So like in this, let's just look at parrots, for example, this gray one here. So in this case, there's a red square in the bottom right. So that means there's a shift in exons on that branch. And so this basically goes through the tree. So, but the main sort of bottom line here is that we detect 15 of these model shifts within a 5 million year interval on this phylogenetic framework. So that's quite a remarkable pattern that's unlikely to be a coincidence. And another way to look at the actual parameter estimates is to compare the estimated equilibrium base frequencies to the empirical base frequencies estimated for each partition. So in this case, so for each taxon partition, so for example, if you just focus on exons over here, this is showing you that for each model shift, the deviation between the empirical, so the raw proportions of equilibrium of base frequencies in a particular group is very different from the estimated equilibrium base frequencies. So so basically the height of the magnitude of the bar basically just tells you something about the magnitude of the implied or inferred difference. And these patterns are most pronounced in exons. There's smallest in introns, then UTRs are bigger, and mitochondrial DNA are close to exons. So I have some ideas about what's going on here, but we can move on and return to it. So this is clearly this seems like there's maybe something going on in this part of the tree, right? Like it doesn't that's sort of self-evident, but we can make this a little more quantitative and model the probability of a model shift as a function of time distance to the boundary itself. And when we do that in a logistic regression framework, we can do a couple of interesting things. But the main sort of takeaway from this is that you read this from this side, from the right side to the left side, and as you go in this direction, you're approaching the KPG boundary. And these curves represent the sort of the probability of model shifts in each of these different data types. So if you follow the black line, this is sort of the aggregated signal across different data types. We can estimate that the probability of model shifts at the time of the oldest inferred shift is about 50%, while also accounting for non-independence due to phylogeny. And doing it in this framework is convenient because we can also then explore things like, are there any other quantitative features that covary with these patterns of model shifts? And one in particular that we were interested in checking was this idea of phylogenomic discordance, because we know a priori that this is likely to it also increases towards the boundary itself. And when we include this in the model, it turns out that this doesn't really end up being significant. So we argue in the paper that this is not likely to be a confounding factor. But anyway, so what's happening to like the birds, right? So like there are these interesting model shifts, but what is happening to their biology? So in order to answer this, we took from this stochastic process modeling approach that I told you about at the very beginning, and applied a method called L1OU to look at 8 different life history traits. And so here's the list here. They're basically, we basically picked all the traits that we could find data for. It was sort of like it was an agnostic kind of targeting. But the idea is, with this approach, is that it allows you to fit this bounded evolution model, but with with one more parameter, this thing called theta, that describes the evolutionary optimum of a particular trait's value through time. So in this case, we're going to estimate this parameter called theta and see if that parameter changes across these different groups that are identified, but by molecular data. Is everyone sort of following, more or less? Okay. So the hypothesis that we're trying to test is there evidence that there are changes in these life history parameters that are coincident with the positions on the tree where we identify molecular shifts? So when we do that, we find that in most cases, so every time you see, for these plots, every time you see a light gray bar that's higher than the dark bar, that means that there's more evidence in favor of a shift in these life history characters relative to sort of a false positive rate for the method itself under an OU model. And so under 2 different sort of information criteria, we find that we basically find that we have very high support for the hypothesis that there are sort of shifts in these life history characters associated with molecular model shifts. So which of those traits is most interesting? Which of those traits is like most closely associated with molecular model shifts as we identify them? So the approach that we're now going to use is we're going to draw from machine learning. We're going to use this approach called the random forest classifier. And we're going to, because this approach is so flexible, we can now sort of increase the scope of the characters that we're looking at. And so instead of looking at 8 characters, we're now going to look at 24 life history characters. And we're going to ask which of those 24 best predicts where you see these model shifts on the tree. So when you do that, this is a plot of the variable importance. And at the top here are really is where most of the action is. And it turns out that really 2 or 3, most of the variance in where these model shifts are can be explained by just 2 or 3 life history characters. I'll go into what they are in a moment. But the bottom line with this random forest classifier is that it does an extremely good job of predicting where these shifts are occurring. And it turns out that the number 1 predictor of these model shifts is this thing called developmental mode. And if you know about birds or ornithology, you'll know that developmental mode is this critical axis of life history variation in birds that describes sort of how altricial or precocial you are when you hatch, so like how embryonic you are. So birds are super variable in this. Some birds are basically ready to go and like can fly within minutes or hours, and some birds are very, very embryonic when they're born or when they hatch. So this comes out as by far the best predictor, followed by adult body mass, and then generation length is a little bit less. So I should also emphasize that this random forest approach is not a mechanistic model. It's sort of hypothesis generating more than it is hypothesis testing, but it allows you to sort of rank and compare both quantitative and discrete characters in the same framework. And so I think it's a really valuable tool to sort of trying to get at what might be going on here. So molecular model shifts can be predicted by shifts in developmental mode or in body mass. But let's ask another question, a related question. Do those characters change in a consistent direction with molecular model shifts? So do molecular model shifts predict shifts in these phenotypic characters in a consistent way. So I'm next going to show you a bunch of plots that look kind of like this, that have these distributions that reflect estimates of this parameter theta that I talked about a few minutes ago. This is estimates of the evolutionary optimum of the trait value associated with molecular model shifts. So we're running a model with this free parameter theta, but we're running it so that the model can shift at the points where we identify these molecular shifts. And so then we're going to estimate this theta parameter to see how different it is and which direction they're shifting in. So when we do this for the top two ranked traits for developmental mode and body mass, we find that in most cases, these molecular model shifts reflect decreases in these parameters. So in this, if you read this from the bottom to the top, here's sort of the ancestral regime. And then as you go up through this, you'll basically, the thing you want to do is sort of compare where the mode of the distribution is relative to the mode of the ancestral distribution. And so in most of these cases, you'll see that these are to the left of the ancestral distribution. And so if you dive into this a little bit more deeply, you find that in almost every case, these shifts represent derived decreases in the trait optimum relative to a given ancestor. So this is pretty cool. So this is, I mean, it's not perfect, but like broadly, it seems like these molecular model shifts are identifying shifts towards increased altriciality. So in this developmental mode, so increased embryonicness when you hatch, and decreased adult body mass. So this is consistent with this other idea that I explored previously in my PhD called the Lilliput effect, which is this idea that mass extinctions are often associated with size decreases. So but in this case, we're identifying, this is entirely derived from a model which is identifying genomic changes, which we're then using as anchors to look at phenotypic changes. And this is sort of supporting that hypothesis. Okay, so another thing, there's a lot. So another thing. So we also got really interested in whether or not avian physiology might be responding to these molecular model shifts, or these molecular model shifts might be indicative of avian physiology in some way. So the way we addressed that was by looking at basically patterns of metabolic scaling. So there's this law called Kleiber's law that describes the relationship between basal metabolic rate and body mass, and this is sort of one of these like universal like laws in biology, which are unusual. But basically, the idea is that metabolic rate scales to the 3/4 power of body mass, broadly speaking. Not perfectly. So here's this picture that I got from some guy's Twitter account. It's actually not published, but it's so cool I had to share it. It's like looking across all these different groups and showing that this pattern basically holds across a huge taxonomic range. So birds are a little bit offset from the rest, but they're still sort of following the general trend. But for us, we want to ask, basically, are there, like what we asked for using this model of shifting theta, is there evidence of shifting allometry? So shifting slope or intercept in this allometric scaling relationship, which is also associated or not with these molecular model shifts. So we're again going to ask, is there evidence of a shifting macroevolutionary pattern in avian physiology now associated with molecular model shifts? To do this, we're going to use a tool called Bayou 2.0, developed by Josef Ueda. The paper was published in 2017 describing how this works. But they previously investigated this same hypothesis that the allometric scaling relationship between basal metabolic rate and body mass may have shifted across the vertebrate tree of life. And they basically found that these shifts do occur, but they're very rare. But the taxonomic scale that they looked at didn't really allow them to look at fine scale variation within clades. And so we wanted to do this, but apply it to this bird data set, but again, ask this question in the context of these molecular model shifts. So we're going to ask, at the points where we're identifying molecular model shifts, is there evidence of changes in the allometric scaling relationship, right? So instead of showing you a plot like this, which shows you this sort of the scatter plot, I'm going to show you a plot that basically plots the slope and intercept separately, so you can sort of get a better view of what actually is going on. So on the top here, we're plotting the allometric slope, so the slope of the line, and then the bottom, the allometric intercept. So our prior expectation that we used for this analysis was that basically that metabolism would follow this Kleiber's law, this 3/4 scaling law. But when we run this model, we find that the variation is a little bit more pronounced than that across birds. It varies from about 2/3 to 4/5. The main thing that's striking, though, about this with respect to the models, with the molecular model shifts, is that oops, no, that this group in particular, this group called Passeria, which is a really ancient group within neoaves, basically represents a shift towards smaller body sizes within bird evolution. And with this group, you see this big jump or big decrease in the metabolic scaling exponent and an increase in the intercept. So this means that the line is going like steeper to like shallower in this particular group. And so just a few weeks ago, there was a paper published in Science describing this pattern exactly, basically, and found that it basically developed a new kind of theory that suggests that the whole phenomenon of elementary scaling is a product of life history optimization and not actually constraints. But they basically predict that evolutionary shifts towards smaller sizes are expected to have smaller scaling exponents. So by anchoring this model or this approach with molecular model shifts, we're able to identify this pattern of basically a shift towards a decrease in scaling exponent, which is now we can sort of predict from theory, which is pretty cool. Okay, so wrapping up, was the mode of genome evolution associated with the end Cretaceous mass extinction? I think we have good evidence that it was. The approaches that we've used are pretty new, and we're still exploring them in many different ways. But everything we've done is consistent with this hypothesis. Nothing has rejected it. And were these shifts in genome evolution associated with changes in life history or physiology? Again, I think we have good evidence that this is the case. And it illustrates this really deep connection between genome evolution, life history evolution, and Earth's history. And I know the bird people will laugh at this, but I think that we can think about the mode of genome evolution as a sort of a new axis of life history variation, because it is so tightly linked to these different regimes of life history variation that we're able to detect. And so given that, I think we can sort of conclude that this represents, in much the same way that Simpson talked about quantum evolution associated with mass extinctions or cataclysmic events in evolutionary history, this does look like that kind of a pattern, where we're seeing this early burst of disparity followed by basically evolutionary stasis over long periods of time. Okay, so with that, I'm finished a little bit early, but I can take questions or I can go into more detail about anything we've talked about. There's a lot that I just threw at you. I hope it was sort of understandable. But yeah, any questions? Go for it.
Beyond like the association between the mode shifts and life history, do you have any ideas about why changing life history optimization might change the modes? Like to be specific, like a lot of the modes that are changing are, for example, the equilibrium frequency of C bases.
Right.
So what are some hypotheses for the connection there?
Yeah. So I didn't go into this at all, but the main one that I'm thinking about, it has to do with something called GC bias gene conversion. So if we go back to this one, the exon data, and we look a little bit more closely at this radar plot, you'll basically see that most of the variation is sort of happening on this axis, so like the GCAT axis. And so there's some ideas that connect this phenomenon called GC bias gene conversion to the life history spectrum. And it has to do with basically the effective population size. It has to do with basically a mutation bias that preferentially basically replaces mutations with G's and C's as DNA is repaired. And so there's some reason to think that might give us a more mechanistic link to why this kind of molecular evolution is indicative of shifts in life history. But as for the other data types, I'm not sure. I mean, it could be that those processes are operating across the genome, or it could be that. But I mean, I guess I would say that given this pattern, where this is very clearly showing us that the signal of these model shifts, the magnitude of these shifts relative to the empirical data is really different across these different data types. So I would interpret that as evidence of different, sort of sub processes that are managing or mediating these different patterns. I don't think it's a coincidence that exons are showing the most action, because exons are transcriptionally active, and there's some evidence to suggest that transcriptionally active regions are more subject to this GC bias gene conversion phenomenon. But why we're seeing shifts of similar magnitude in the mitochondrial DNA, which is theoretically non-recombining, I don't know. Yeah, so there's clearly a lot that we have to learn about what the mechanism of this might be. I think the main point of this project was to sort of see if we can identify where these shifts are and whether or not there's any evidence that they're associated with the boundary itself. And I think actually diving deeper into the mechanism is going to come later. Yeah. Yeah, go for it.
I had a couple questions for methodological. One of them was you had a little lack of congruence with the AIC, I think it's BIC
Sure.
metrics.
Yeah, I'll go back to that.
When I see that, that gives me, it's pretty congruent, but you notice like a dip within the.
Yeah, it's a good point. So this.
How do you?
Yeah, so I didn't go into this at all. But this AICC criteria is easier than this BIC criteria. So it's just more conservative, basically down here. So I think what's happening is that this is just this is this phylogenetic BIC that was proposed in the L1OU paper. So it's sort of its own thing. But if you look at the sort of average, so there are two groups here that basically were, so it's essentially congruent except for these two, I think. Yeah, and then there's,
the one that jumps out,
which one?
The passerine or whatever, the P in the middle.
This guy, Pasteuri? Oh yeah, this one. So Pasteuri is it is curious how that's so, it's basically the opposite signal. I can't explain why that's different. It's just that it's a totally different, this BIC criteria, this phylogenetic BIC is calculated in a very fundamentally different way than this is, so that could explain it, but it's, interestingly though, however, this is the only, so there are two shifts on the tree that we detect that are like obviously not KPG associated, and this is one of them. So this Pasteuri is more derived. So I'll go a little bit. I'll show you where it is. So in this case, we detect. So Pasteuri is here, this one here. So there's this one, and then there's this one down here at the root of the stem of Neognathle, this group here. And so I don't think that these are KPG associated, but everything else is within this sort of tight 5 million year interval, at least relative to this phylogenetic framework. Anyway, so I don't have a good answer as to why that one is totally different. But my interpretation is that it's just it's a different criteria, so it's calculating, it's weighting it in a different way.
I mean, it is, but
Yeah.
I do a lot of these similar comparisons, not this type of.
Sure.
When you see disparities, even if it is a different criteria, it's like, well, it does say something, and I'm always curious what that is. So.
Yeah. No, I totally agree. I don't know the answer. Yeah.
And I was curious, the metabolic thing was pretty interesting, but so why don't you go to that plot with the metabolic where you created those biological plots?
Yeah.
So I see the prior line. Did you set these priors?
Yes.
So why would you do that? Why would you not leave it open?
Because it's a Bayesian analysis, so you have to give it a prior for the parameters that we're interested in. So
you can make it wide open, or you can set it and give it a constraint.
Yeah. So this is
based on. If you're interested in what the answer is, why would you constrain it towards what the theory already says?
Because we're interested in the deviation relative to the expectation. So the expectation that we have from basically the theory of whatever it's called, the metabolic theory of ecology, is that metabolic rate scales to the 3/4 power, and so that's our prior expectation. So we didn't fit it as like a strict line. It's a distribution. I forget what the variance is that we set around that, but it wasn't like a set line. It was a distribution of but that's just the nature of Bayesian analysis. You have to give it a prior that describes your sort of prior expectation.
Yeah, I understand that.
Yeah.
I guess it depends on your question, but if your question is what is the slope, then setting the prior to that, having that constraint around that is informing it.
Sure.
And that seems curious. If you're actually just curious what the slope is, then why would you just let it be? Why would you let it be in constraint? I mean, you're biasing your analysis, which is fair, and I understand Bayesian quite well. But
Yeah.
And that makes tons of sense at certain times. But if you're actually trying to understand what that slope value is, why would you constrain it up front based on theory? You're just testing the theory and saying, what is the slope? Then you let the Bayesian analysis do its thing, wide up the priors and see where it goes.
I could certainly run it again with a flat prior.
It's the same thing.
Yeah.
Just,
Yeah.
you understand my point?
Yeah, I do. Yeah. No, I know. It's a fair point. I guess I'm not super worried about it because the question is more about what is the deviation relative to the prior that we're giving it. And in this case, we're looking at, basically we're finding these like the thing, so these analyses are definitely more ambiguous than the other stuff, for sure. and the posterior, so these are the posterior distributions. So in some cases, they're extremely wide. And so the signal of what these allometric relationships is weaker than the signal of these like allometric, than these sort of shifts in theta. So it is a weaker result for sure. But yeah, the idea, we certainly could run it with a flat prior. We didn't, basically because that's how the paper that published it did this. So we followed their prior sort of as a template. And they were also investigating metabolic scaling, so we figured that was an okay prior to use. But it's a fair point. Sure. I mean, it certainly could be biasing the results in some way. Yeah.
Strange assumption. I haven't read that paper yet, so
Yeah, go for it.
I guess I have a couple of questions. Mostly motivated by the fact I'm incredibly curious as to how is it that you end up with this really monophyletic or nearly monophyletic
Sure.
regime shifts.
Yeah.
All right at the KT.
Yeah.
So I was wondering if you have some speculation as to how you end up with such clean splitting of evolutionary regimes, but also how did you do that in the sense of accounting for uncertainty in time estimates? Like, is there a particular distribution on the aging of the?
So I'd be very happy to send you my draft of the whole thing. You can read all about it. But there's a whole, yeah. So there's great uncertainty in the timing estimates. But the approach that we took with this was basically sort of something that I alluded to at the beginning, which was to say we're just gonna take what the fossil record tells us, and we're gonna assume that that is the story of bird evolution, because redoing this on a range of divergence time estimates was sort of it would magnify the amount of work 100-fold, and so we didn't do that for this project. But the thing to keep in mind is that when you run this method, this method, you're running it on a tree that looks on a phylogram. So the method has no information about time when you're actually running it. It's running on branch links that are in substitution units, and so then the results that I showed you are then projected onto this previously published onto this tree, and we're assuming that tree is a fair tree to use. So that's an assumption of the work. Yeah, but you're right. There is great uncertainty in these divergence times, and so the paper that I mentioned before, we talked about that a lot. This paper that we published in Systematic Biology a couple of years ago, that was the focus of some other work. I don't mean to dodge your question, but basically the approach we took was to just ignore that part of the variance for this particular project, just because it would have added too much. It would have been. It's kind of a different question. So I think that so for example, in my view, the fact that we are identifying, so we're getting in a tree that looks like this, and yet when you reproject that onto a tree that's time calibrated, you find all these shifts within this tight interval. That in and of itself, to me, I think that is evidence that the tree is more or less accurate, because the only explanation for the tight congruence, this tight interval, is that it has to do something. It has to do with the rapid diversification of birds at the end Cretaceous. That's my take on it. It could be that, I mean, if that wasn't the case, then that would imply that that these divergence times, if you extended the tree, 100 million years, then they'd be much more spread out. Of course.
Yeah, the other thought that I had was
Yeah.
that if you really have sudden extinction at that scale, maybe all this is just a random sorting of regimes that were otherwise
It could be.
much less discrete. In the previous phylogen, but removing uncertainty sort of also, I think, might limit the ability to test that, especially
Yeah.
trusting the fossil record at face value, knowing that taphonomic preservation
Sure.
can introduce them.
Yeah, no, you're absolutely right. You're right. That is absolutely an element of this that we haven't dealt with. So it's a fair point. Yeah, go for it.
Yeah, that was really interesting. My question is kind of a combination of Teresa's and Hernan's.
Mm-hmm.
And I think I'm just having trouble piecing together the direction of causality between all these different bits. So there's like the KPG boundary, there's sort of mass extinction, and or subsequent diversification of birds following the KPG boundary. You're seeing a shift in equilibrium base frequencies associated with clades that are of that age.
Mhm.
And you're also seeing a shift in some morphology traits that you're looking at.
Right.
But like, what is causing what? I mean, that
I don't know. Yeah,
As,
that's a good
as,
question.
as you're presenting it, it's
Yeah.
just like a really interesting, like,
Yeah.
wow, all this stuff happened at the same time.
Yeah.
But it seems like at different points in the talk you were implying different directions of causality.
Okay.
And I'm curious about, like, is the whatever GC bias gene conversion the mechanism by which you see accelerated evolution in these clades following the KPG boundary, which is what led to diversification rate shifts? Or
Yeah.
is it like something completely different?
That's a great question. I don't know. I think that the way that I think about it is more in the context of the end Cretaceous acting as a massive selective agent on phenotypes, and that phenotypes in life history are related to how the genome evolves. And so I think that the direction of causality that I'm thinking is more that direction. But as to how the mode of genome evolution changes, and then that might impact how that clade can evolve, it could be both directions. So the answer is maybe both. But I don't really know. Sorry, go ahead.
I think the part that I'm having the hardest time connecting the dots between is the mode of genome evolution and sort of impacts of that. I don't really understand, beyond the potential, implication of GC bias gene conversion, I don't really understand how you're connecting the dots between different equilibrium base frequencies inferred for these different subtrees
Mhm.
and anything else biological.
So I'm sorry if I wasn't clear about it, but the way we're studying these basically changes in phenotype is by taking the locations of the model shifts and then using that as like an anchor point.
I got the modeling part of it. I'm
Okay.
saying, like, biologically, what's the connection between a shift in the equilibrium base frequencies on a portion of the tree
Sure.
and anything else?
Yeah. Well, I kind of alluded to one idea when I answered Teresa's question, which is this idea of like GC bias gene conversion. So there is some reason to expect that mechanism could be related to the sort of fast flow life history spectrum. But my sense is that we really don't have a very good intuition for how those parameters, the rate matrix or the equilibrium base frequencies, may directly, what the mechanism is connecting to phenotypes. I think we have a much better intuition for like rates of molecular evolution. So for example, like it's kind of intuitive that you would think, okay, you have a shorter generation length, so the rate of molecular evolution is faster because you have faster turnover. That kind of intuition doesn't really exist for these kinds of shifts. As far,
GC bias gene conversion, you should expect it to show up also in the rates of molecular substitution.
Yeah, so potentially. So we haven't really looked at that directly, but that's another separate thing, I think. Yeah, it's a great question. Yeah.
Fun talk.
Yeah. And sorry, I don't know who was
Just
next.
Gideon made me think really quickly. Would it make sense to repeat the analysis with amino acids to see if all those changes in
Sure.
base composition cause codon biases or not? Because maybe it doesn't make any difference.
Hold. I've done that. Or not exactly that, but something similar. So we looked at codon, basically patterns of codon usage, and it turns out that the short answer is that yes, there's differences in patterns of codon usage across these, at least in the exon data. That's the short answer, but we can talk more about it. Yeah. I think you may have had your hand up first. Go ahead, go for it.
I'll just pitch you an easy one. Can you go back to your violin plot?
Sure.
I think I just missed the specific link between these distributions and the statement at the top that the shifts are associated with molecular
Oh,
shifts.
yeah.
Where
So
does that come in? Is it just the clades that are being pictured?
yeah, so there is a there's like a posterior probability for the shifts that I haven't shown. But the thing that I'm trying to emphasize are the parameter estimates. So each time you see one of these violin plots, that is a group that is identified on the basis of one of these molecular model shifts. And so the fact that we see these differences in parameters is indicative of this idea that the scaling relationship may have shifted in association with these molecular model shifts.
So would we expect this distribution of shifts in parameters to be any different than if you picked any other place randomly on the tree?
I mean, I think you would, because it's a different configuration
You can
of
test
points on
it.
the tree. Yeah, certainly you could test it. Yeah, certainly. Yeah, certainly. Yeah, go for it.
Yeah, there's a question in the chat from Bruce Martin.
Okay.
Really cool talk. Did you investigate whether transition rate parameters also vary across the tree in addition to equilibrium?
Yeah. So the short answer is we were initially doing that, but we basically found that they were only changing very, very slightly across these different regimes. And so for computational reasons, we decided to just focus on equilibrium-based frequencies because we ended up having to run these analyses like hundreds of times. And if we also included the rate matrix as a set of free parameters, then it made the runtimes like infinitely longer. So we basically decided for computational reasons to focus on the equilibrium-based frequencies. Yeah, but that's a great question, and also a super interesting axis of variation here that we are basically holding constant for this particular analysis. Yeah, Jeff.
Can you go back to the Kimball et al. Curious how a process of going from a super tree,
Sure.
right? There's no data there.
Sure. Well, this is
data or matrix topologies of other analyses.
Sure.
And extracting a data set from that, and whether that data set reproduces the same tree or.
Yeah. So this is a really interesting paper, but basically they took. There are some data behind this. So it's they basically took as you know the phylogeny of birds is like uncertain, especially of this deep part of the tree, and so their approach was basically to take a number of these different sort of genomics estimates of bird phylogeny and to weight them in proportion to the strength of their signal, and then basically assemble this super tree from that. But then they layered in on top of that. I think about 1,000 genera based on mitochondrial DNA. So it's kind of a combination of both approaches. And the reason I wanted to use this instead of trying to re-estimate the tree on this new data set was because two reasons. One, I wanted to sort of strategically avoid getting into that question of like what the bird phylogeny is and what the topology is, because it's a very difficult problem. And then secondarily, we thought that maybe it would avoid any sort of accusation of circularity in review, because if you are inferring a tree using a particular data set, and then you're then using that same tree to estimate sort of parameters of molecular evolution, there may be some sort of circularity there, and so we kind of wanted sort of a sort of pseudo-independent estimate of the tree to then look at patterns of molecular evolution on. Does that sort of answer your question?
Yeah, I guess I was kind of following up on whether the data set that you extracted and then kind of amplified, whether that reproduces a topology that looks like what you're actually mapping those kind of characters onto.
Yeah, so that's a really interesting question. So I thought about this. So there is a real possibility that some of the clades we're identifying as having model shifts exist or were recovered in the first place because of convergent patterns of molecular evolution that have sort of sucked them together in a place of the bird phylogeny where there's extremely weak phylogenetic signal. And so it's possible that some of the groups we're identifying may be indicative of artifacts in the topology. That's totally possible, but it's a separate sort of research question. Yeah. Does that get at what you're getting at, kind of, or? Yeah. Anybody else? I think we're just about over with time. Cool. Yeah, thanks.